So my title mentioned metagenomics. If you don't know what that is, I'll give an umbrella term, which just for this talk, this is very argued about. I want to say that metagenomics comes under a wider umbrella term called metaomics, which is a series of methods which aims to analyze multiple species in a sample. It's not like NF-COSERIC, which does boring one genome. Metagenomics tries to analyze multiple at once, but also with multiple molecule types, whether that's DNA with genomics or proteomics and things like this. And many of the methods have come out of microbial ecology. So about 30 years ago, it became very, very obvious that you can't really analyze a particular microorganism in isolation. So microbes are absolutely everywhere. To make you squirm a little bit, there are microorganisms on the seat you're sitting on the ceilings, on the walls, on your skin, inside your body. And they play a really fundamental role in pretty much everything, whether that's making you sick, but keeping you healthy, making your food, cycling nutrients in the soil and things like this. But it became very, very obvious, like I said, that you always have to understand these microbes in the context of all of their friends around them, how they are interacting with each other, and things like this. There we go. And so, as you can imagine, this means that we have to work at scale. So just in your mouth, there's probably 700 different species of bacteria living there at the moment. And increasingly, particularly over the last sort of 10, 15, 20 years, now we've got NGS sequencing, the number of organisms we can study at any one point in any study is going crazy. So every point that you can sort of see, it's so dense you can't really see it, but points here, also with the lines, these represent a different microbial organism. There we go. And this is also reflected in just the sheer scale of number of sequences on big genomic databases like NCBI-RefSeq. This is just bacteria, but there's 1.5 billion bacterial reference genomes there. And theoretically, in metagenomics, you'd want to compare your DNA sequences against all of these. Okay. Also reflected, if you look at NF-core, of the top 15 most popular top pipelines based on GitHub stars, 1/3 of these are directly related to metaomics, and there's a couple more extra which you could also apply to similar data. So really, Nextflow is really great and really popular in microbial ecology, or metaomics in general, because of this sort of ability to scale and parallelize very, very, very efficiently. This is partly also reflected by just the diverse number of researchers working in this area. So you have scientists trying to look for new biomolecules, new drugs, things like this, or precursor molecules to these from these microorganisms. You have clinicians trying to identify what is making you sick, but also keep you healthy with all the bacteria in your gut. It's also outside of the hard sciences, but with even social scientists, which is more where I'm coming from. Lots of people are interested in the microorganisms involved in fermentation and how this is passed through families, in this case, for example, Mongolia, because literally you have like heirlooms in terms of the starter culture that produce these things. And I actually come originally out of ancient DNA, and we apply all of these methods also onto ancient stuff. So trying to find out what killed people 400, 500, thousands of years ago, you can really apply to all of these. So lots of different researchers working in this area. So for the middle part of this talk, I want to give 3 examples of how you can sort of apply different NF-core pipelines to major aspects or sort of points or milestones of a particular metagenomic study. The first question or problem you often have is just who is there? Where has the DNA in your sample come from? And like I said, we want to identify all the hundreds of thousands of bacteria in there. And there are a series of approaches who try to do this very efficiently with taxonomic, which fall under the name of taxonomic profiling. In NF-core, and I hope you like metro maps, you can see quite a few of these. NF-core tax profiler addresses this problem, so it allows you to take your DNA reads and run many, many different taxonomic profiling tools and databases, critically, to solve this problem. The reason why we need this is there is no actual gold standard tool. You might hear Kraken, for example. That is not actually the best in all cases. And also database choice is very important in this thing. So we use Nextflow to basically parallelize this, and also develop tools with my colleagues to standardize the heterogeneous output to make it easier to make consensus sequences, so sort of, well, not consensus sequence, but a consensus call of what's the most likely bacteria you have in there. Once you've done this, the next question is, what about the unknowns? What if you don't have the reference genome in public databases of the bacteria that might be in your sample? SARS-CoV-2 is a good example of this. It's a novel strain. For this, Metagenomics, we have metagenomic de novo assembly. In NF-core, we have a pipeline called NF-core-MAG that sort of wraps around this, which does this de novo assembly, but also taxonomically classifies and say what most likely that sort of species your genome is most similar to, do annotation and quality control things against standards. The pipeline supports both short reads and hybrid assembly, so if you have long reads that you can sort of help scaffold this. And also ancient DNA has a dedicated ancient DNA mode, which the approaches were applied to actually reconstruct novel species, that strain, from about 11,000 years ago, which is pretty cool. And finally, probably in the last 10, 15 years, I would say, what's really starting to shift, in particular in the microbiome research world, is we don't care so much about who is there in terms of the species of bacteria or microorganisms you're finding, but it's more about what are they doing. But obviously, the biologists in the room will know there are many, many different types of genes and very interesting things. But in many cases, these studies or tools often do this on a one-per-one basis. So then in NF-core, we have NF-core FuncScan, which again parallelizes this. This is why Nextflow is really, 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 really cool. And we can run all of these tools in parallel in a really, really efficient manner. Whether that's in FuncScan specifically, you're looking for antimicrobial resistance genes, so maybe your clinician, you want to know, is there a risk of antibiotic resistance? But also biosynthetic gene clusters, so if you want to try and identify maybe drug precursors for new antibiotics and so on. But that is not everything. Again, under the general term of metaomics that I've defined for this talk, there are many, many different pipelines in NF-core that are all very complementary, and you can really go from very early data processing, data downloading, all the way to secondary analysis where you start homing in on particular, whether it's viruses or phages and things like this, building trees and so on. But with this complexity, as you see from this metro map, the question becomes in coordination. So how can we make sure that we work in almost a seamless approach when you're doing a metagenomic project? So NF-core recently set up this initiative called Special Interest Groups, and we have made one called Metaomics, which is designed for both users and developers working in anything from amplicon sequencing to metagenomics, metatranscriptomics, and metaproteomics to come together and sort of make the experience of these pipelines even better. So for developers, we have sort of objectives of trying to reduce redundancy. So rather than doing the typical bioinformatician thing of reinventing the wheel 40 billion times, we want to try and coordinate to make sure one pipeline does the one thing best, and then, works with the next one. We also want to coordinate long-term development. So I'm calling this the Wikipedia rockstar plot. If you're familiar with the band member timelines, I've done this for programmers. And MAG is a really good example of this, how within NF-core we've been able to actually pass on the maintenance and development of this pipeline over a much longer time period, so this is 6 years, than most bioinformatic pipelines or tools which die once the person graduates from the PhD. So this is really, really nice to see. And we also want to start going in the direction of this for meta pipelines with pipeline chaining for improved compatibility to make, again, it much faster to go from each stage of these projects. And in fact, over the last few weeks, we've already had within the Special Interest Group our first sort of mini hackathon, where this slide is actually out of date because we had more development during the NF-core hackathon the last couple of days. But we can now, in development branch, releases, and a couple of feature branches, go from right at the beginning all the way to this end point that we currently have, where each pipeline can produce a sample sheet, so with the list of paths and the relevant files needed for the next pipeline to execute. So you can almost just go run one pipeline and execute the next pipeline without very minimal sort of interaction to make sure it sort of executes, which is really, really exciting. And I'm hoping this really goes much further and deeper as within NF-core, because there's a lot of interest in such a topic. For users, we want to promote and assist the adoption of NF-core pipelines. Another thing that I think a lot of bioinformaticians, particularly in academia, don't do very well is actually advertising and getting users to come and use their pipelines. So we want to try and do this for NF Core. We also want to improve documentation with better tutorials, maybe on specific contexts as well. You know, maybe saying, okay, if you're working in viral metagenomics XYZ and you want to do these steps, maybe run these pipelines with these settings to help guide people and sort of ease people into these very, very powerful pipelines, but sometimes quite complicated pipelines that we often get in NF Core. And we also want to maybe coordinate to standardize parameters across pipelines. So in NF Core, we already have these beautiful interfaces which are very consistent across each pipeline, but sometimes which flags you turn on and off, or like how you define particular paths, becomes a bit blurry. So maybe we can try and make this easier for users to switch between pipelines and know what they're providing. There we go. If you're interested in Metagenomics in general, we have our page on the NF Core website for Metagenomics. We have a dedicated Slack channel. We also have monthly meetings currently, a bit European focused, but we are open to having extra meetings or shifting the time if you're sort of in a different time zone. And if you're outside of the sort of special interest group, you're interested in these pipelines or want to help sort of push forward the development of these, please come speak to myself or Daniel Lunden, who is somewhere in the crowd, who's the other member of the leadership team here at the summit. There you go. Okay, so to summarize, Nextflow is great for Metagenomics. We really need to scale because of the sheer number of genomes that we have to compare our DNA against. And this applies to Metagenomics in general, whether that's metaproteomes and so on. NF Core offers multiple powerful pipelines for this as well. I gave 3 examples just within metagenomics, but there, like I said, one of the pipeline maps, there's many different options here. And our special interest group, we hope to strengthen coordination and really ease both the experience of developers and users for using these. And this is my acknowledgement slide. So I just want to thank particularly all of the co-authors who are not everybody's contributed to my pipelines that I've presented today, but these are the core people. The NF Core community is a wonderful place. Everyone helps in all over the place. It's wonderful having people coming up during the hackathon today who work in a completely different group saying, I'm making a module. I was like, I can use that. That's great. And otherwise, obviously, of course, thank you to my employers, the HKI and MPI-EVA in Leipzig, the funder, the Werner Siemens Stiftung, and the Nextflow Ambassador Program for helping me be here today. So thank you very much.